Hi everyone, I'm Emily and my group members are Mackenzie, Melissa, and William. We are presenting on spatial temporal control of biochemical cues through the article called A Photoreversible Protein Patterning Approach for Guiding Stem Cell Fate in Three-Dimensional Gels. In vitro platforms commonly used lack the dynamic heterogeneity of in vivo tissue. In vitro platforms that are able to capture this dynamic heterogeneity are essential for the future of tissue engineering. The type of chemistry used in this paper is the first use for the controlled immobilization of full-length proteins. This paper exploits two bioorthogonal photochemistries to reversibly immobilize bioactive proteins with spatial and temporal control in a synthetic cell-laden biomimetic scaffold. For more insight, bioorthogonal refers to chemical reactions that can occur inside living systems without interfering with native biochemical processes. These reactions are designed to be selective and specific, meaning they can only react with particular chemical groups or molecules within the cell. The methodology used in this article will prove useful to understand how cells respond to extracellular cues and for engineering materials for regenerative medicine. In work that has been done previously, we know that hydrogels are currently used as in vitro 3D cell culture platforms. They are static platforms that can't mimic spatial and temporal changes that are prevalent in vivo tissues. So to mimic a dynamic tissue environment, researchers have used photopatterning, which involves using light and a photoinitiator to initiate a reaction and create patterns or structures within the hydrogel. The photopatterning alters scaffold functionalities in space and time. This is very important to replicate the native physiological environment. Previous studies have utilized photopattern introduction of biochemical signaling molecules into hydrogels to emulate physiological changes in ECM composition. There have been advancements in the introduction and removal of these biological cues through wavelength-specific photocoupling, which is the process of using light to cross-link or polymerize hydrogel precursors into a 3D network structure, and also photocleavage chemistries, which is when light is used to split molecules into smaller pieces. These methods were limited to simple, highly engineered small molecules or peptides.
Thank you so much, Emily. The overall motivation of the study was the ability to recapitulate the dynamic heterogeneity of natural tissue, which is needed for fundamental biological studies, as well for the further development of effective strategies for tissue engineering. Additionally, the researchers wanted to be able to control the presentation of full-length proteins in 3D hydrogel microenvironments, since on most of the past work platforms cannot capture the spatial and temporal variation with which physiological cues are presented to cells in vivo. In terms of the hypothesis, the researchers wanted to know if their approach would be good for attachment and subsequent detachment of full-length proteins with good control in space and time while retaining their bioactivity. They believe that a protein patterning approach could provide further avenues to probe and direct changes in cell physiology in response to dynamic biochemical signaling in a spatio-temporal manner. To touch on some of the design criteria that guided the approach that was taken by the researchers, first, the experiments must allow attachment and subsequent detachment of full-length proteins with good control in space and time. Second, they needed to make sure that all the chemistries must be bioorthogonal and cytocompatible in order to achieve the specific bioconjugation necessary for working with fragile proteins in live cells. And finally, the patterning must be executed on timescales that are relevant to the biological functions of interest. The general strategy that the researchers followed was a combination of three bioorthogonal chemistries that enables reversible pattern presentation of full-length proteins within polymeric hydrogels. They utilize the strain-promoted azide-alkyne cycloaddition reaction for network formation, a photo-deprotection oxime ligation sequence for protein introduction, and an ortho-nitrobenzyl ester photoscission reaction for subsequent protein removal.
Thank you, Melissa. In order to create this spatio-temporally controlled gel, multiple chemistries were combined and considered. To create the network which provides mechanical support and body of the scaffold, they utilized 4-arm PEG and an MMP degradable crosslinker. The 4-arm PEG is functionalized with a cycloalkyne, which can react via click chemistry with the MMP degradable crosslinker, which is homobifunctionalized with azide groups. Via the same click chemistry, the cycloalkyne groups on the PEG react with the azide groups on an NPPOC photocaged alkoxyamine. The purpose of this will be discussed shortly. This gelation occurs through strain-promoted azide alkyne cycloaddition, which means that the alkyne ring is stressed, making its reaction with an azide spontaneous. The aforementioned NPPOC caged alkoxyamines distributed uniformly throughout the hydrogel, which is R1 on the picture on the right, undergoes irreversible beta elimination when exposed to ultraviolet light. This is the uncaging that the article mentions. It allows for protein attachment at these sites throughout the hydrogel. A molecule called NHS-ONB-CHO, which will be referred to as NHS-oxime for our purposes, is also added to the scaffold. Its purpose is to link the protein to the hydrogel at the NPPOC caged alkoxyamine groups. This molecule imparts photopatternability through the introduction of reactive aldehydes linked through photosystyl O-nitrobenzyl ester moieties. It is photopatternable because the groups that are uncaged by the UV light in step 2 can react with the NHS oxime, and the NHS oxime can then react with the protein of interest. Once the scaffold is cross-linked, laden with cells, and functionalized with the protein of interest, this protein can be removed from the scaffold in a controlled manner. This is useful because the protein can be removed from certain parts of the scaffold at different time points to create gradients or change the activity of a certain section of the scaffold. So exposure to UV light causes photoscission of the protein from the scaffold, specifically the O-nitrobenzyl ester moieties, linking the protein of interest, which is R1 in the picture, and the hydrogel, which is R2 in the picture, and these undergo photocleavage upon exposure to ultraviolet light. This releases the protein from the scaffold. And now I will pass it on to Mackenzie.
Thank you, William. So now I'm going to talk about a few of the experiments that they did in this study. The first one was to investigate protein attachment using their strategy. So they put 2 different protein solutions into the hydrogel, one of which was functionalized with aldehydes, which made their attachment not reversible with UV light, and then the other was functionalized with the NHS oxime molecules, as we just discussed. So it was removable using UV light. They used immunohistochemistry to tag the proteins and see where they attached or detached within the hydrogel, and they varied the intensity of the light and exposure time with the removable proteins, which was found to be correlated with the amount of protein attached to the hydrogel. Through this, they established that they can control the attachment of full-length proteins as well as the removal of the proteins by varying the intensity and the exposure time of UV light. They then wanted to figure out if, even though they could control the attachment of the proteins using this approach, if it at all impairs the functionality of the proteins once they're attached. So they tested the bioactivity of 3 different proteins. One was the collagenase enzyme, another was an antibody, and the third was a growth factor. And that they found that the activity of these proteins was not altered after undergoing the NHS oxime molecule functionalization or the gel patterning process. They then wanted to see how human mesenchymal stem cells would respond to spreading cues that they changed over time. So they attached vitronectin to the scaffold using photopatterning, and vitronectin is a protein that promotes cell spreading and has been shown to promote osteogenesis for human mesenchymal stem cells. And they assessed whether osteogenesis was occurring using immunohistochemistry and stained for osteocalcin, which is a marker for osteoblasts. And they found that in areas of the hydrogel that had vitronectin attached using their approach, the cells underwent osteogenesis, while cells in the areas of the gel without vitronectin did not undergo osteogenesis. They also observed that the removal of the vitronectin using UV light caused for cells to dedifferentiate to their original state. So in conclusion, the experiments described here are the first uses of this specific chemistry to immobilize full-length proteins in a reversible manner. According to the researchers, their method provides a strong tool for accurately manipulating the spatial organizations of cells and macromolecules in 3D hydrogels, which may be utilized to generate microenvironments that govern stem cell activity and differentiation. The approach used in this article also has promise to be used in tissue engineering and regenerative medicine applications. Thank you for listening, and we will see you in class to discuss the article further.